channel BioLab, and this is VAMSI. In yesterday's video we have completed about auxins, the physiological effects of auxins, and in this video we will be seeing the physiological effects of gibberellins and cytokinins. Gibberellins and cytokinins. Yes, both will be covering in this video since they are very small and are very little but very important things are to be discussed in today's video so that without any further ado, let's get into the video. And also I have prepared a mind map of the microbes chapter. I will also putting the link of the PDF in the description box. The mind map is for free, absolute 0 rupees, but if you want notes you have to go into the website and pay 5 rupees. Only one time payment, guys. It is only a single time payment of 5 rupees for every notes. Only once, if you pay 5 rupees, every notes you will be getting. Yes, about the mind map, I will be uploading it in the website. There you can go and check it. So also have uploaded a test on microbes chapter. You can go and attend the test fast so that we will be completing the chapter. Yes, I will also discuss some of the MCQs in the while making the session. Soon I will be planning that too, the MCQ series, so that there we can discuss some more MCQs which I haven't included in the tests. So let's start, guys, today's video without any further ado on the physiological effects of gibberellins and cytokinins. Yes, today's video is going to be very important video. You have to make a note of everything of gibberellins and cytokinins since they will be confusing to you. Yes, let's get started. Let's get started with gibberellins. First of all, we have again, like yesterday in auxins, we have only one point, important point about gibberellins. That is, these gibberellins are all acids. Gibberellins are all acids. Remember, Ga we write means gibberellic acid. Remember this. All gibberellins are acids, and till today there are 100 gibberellic acids found, and Ga3 was the first one to be discovered and also mostly found in many plants. Yes, these are the points you have to remember in this, and some more points, important points on gibberellins we have already discovered in the discovery of PGRs. There you can go and check it in the website or in the playlist. It is also available. Please go watch that video and come back to this video. It is just a video before this one. Yes, let's see the physiological effects of gibberellins on plant growth and regulation. Yes, I have already said you that gibberellins are plant growth promoters, PGPs. They promote plant growth. They are PGPs, plant growth promoters. What are these plant growth promoters are also discussed in the discovery video. You can go and watch them. Yes, it's enough for the introduction. Let's move on to the physiological effects. Now, these gibberellins have some important physiological effects, and first one of them is, let's take a different color. Why always? Yes, some ash color. Oh, yes, it happens sometimes. Never mind. Let's move on. These gibberellins have some specific effects on the plant growth and regulation. We'll be seeing one by one. The first one is they increase the length of the plant. Length is increased. By gibberellins. Now, if you abstract gibberellins from Gibberella fusiculae, it was already done by E. Kurosawa, and then if you spray it on any other plant like sugarcane, remember the example, sugarcane, this sugarcane would increase in length, thereby increasing the yield. See, sugarcane, it is like a stick. Yeah, it is like a long stick like this. Think for some time this is sugarcane. Yes, if I do it with brown color, it would not be visible to you. Okay, this is the sugarcane. And what this gibberellic acid does is, if it is sprayed, it increases the length of the sugarcane. If it increases the length of the sugarcane, yield also automatically increases, yield increases. Therefore, gibberellic acid, once it is sprayed, it increases the length of the sugarcane or grape etc. It is used in increasing in length, So that is the first physiological effects. Let's move on. The example is sugarcane. It is one of the most important example. Yeah, I will be soon starting the NCERT line by line series, one shot videos also. If you haven't, no time in this 220 minutes video, there you can go and see 1 hour video full lecture, okay, a full fledged lecture. Now, as they promote plant growth, they delay senescence. Remember like this. As they promote plant growth promoters, growth is promoted, so they delay senescence. What is senescence? It is the olding of plant. Olding means it is the old age of the plant is termed as senescence. A plant getting on by, if you spray this gibberellic acid, plant would delay its age, olding. It delay its olding or aging. Okay, aging of the plant is combated by this gibbsellins or gibberellic acid. Okay, remember this. All the plant growth regulators only concentrate on abscission and senescence. They either delay senescence or delay abscission. That's the second important function or physiological effect of this gibberellic acid. Let's move on to the third one. Yes, hope you are noting it. If you are not noting it in the class, you can go and check out the notes. I have already uploaded this notes on the website. Okay, yeah. These gibbsellins speed up malting process. Sir, what are you saying? You are introducing very new term to us. Is it malting? No, malting means germination. Germination of seed. The germination of seed is fastened by these gibberellic acids. Gibberellic acid fasten germination of seed. Hence, it is used in this malting process. The speeding up this physiological effect is more useful in brewing industry. Yeah, brewing. Maltose. If you ferment maltose, you will get alcohol. Maltose. That one, ragi malt. Please relate yourself to those. The germination process. If it is speeden up, if it is fast enough, then brewing industry is very useful. They can ferment the drink very fast, and it increases the cost. So, cost means the production. This is the third important. The speed of malting. I will be saying a trick to remember this malting and next one also. For malting, remember ragi malt. Okay, for malting, remember ragi malt. For bolting, yes, they promote bolting. What are you telling? Promoting bolting. They help in promoting bolting. What is this bolting now? Is it putting screws, driving screws into the walls? No. Bolting is something that is related to the internodes. See, if this is one node of the stem, this is other node, and this middle portion between two successive nodes is called as an internode. This internode elongation generally happens post flowering. This internode elongation happens post flowering, but when you spray this gibberellic acids, this internode elongates. This internode elongates prior to flowering. Means they speed up the process of internodal elongation, but only in some plants, which you have to remember according to NCERT, that is cabbage, beetroot, and I don't know how to pronounce the third one. I'll be writing the name. First one, example, bolting example, cabbage, beetroot, and rosette plants. Yeah, rosette habit. Remember, the rosette plants, beetroots, cabbages, they promote bolting. Gibbsellins promote bolting in these three plants or three examples, which you have to remember according to your NCERT. That's it with Gibbsellins. I said that I would be saying you something, how to remember these effects of Gibbsellins in malting and bolting processes. For malting, remember ragi malt, and for bolting, remember bolts. Enough. If you remember ragi malt, malting. If you remember bolt, Usain Bolt, bolting, ing. Enough. Those are the physiological effects of Gibbsellins. Completed. Yes, you have to turn this scientific terminology into your basic terminology, then you will be remembering. Do not go for any shortcuts. Remember just like this. Yes, that's it for Gibbsellins. We will be seeing about cytokinins now. We complete successfully Gibbsellins. Now we go for cytokinins. These are very special. Very special. They have specific effects on cytokinesis. That's why they are named cytokinins. Cytokinesis. They have specific effects on cytokinesis of the cell. What is cytokinesis? Maturation of the cell. That's it. Remember it. Maturation of the cell. We'll be reading that also. Cytokinesis. They were first discovered as a kinetic. Yes, they were first discovered as kinetin. We have already completed this in the discovery. Kinetin. But the thing is, this kinetin does not occur naturally. Naturally, it is not found in plants. No. It is found as zeatin. Why zeatin? Zea maize. See, Zea maize is the scientific name of maize, and this zeatin was first identified in the maize plants or corn kernels. That is why it is named as zeatin, and this is so specific efforts on cytokinesis, hence it was named cytokinin. Kinetin is not present naturally. Please remember this. Very important. Then the physiological effects, the important points are very important. Remember this, that kinetin does not occur naturally, but naturally occurring cytokinin is zeatin. It was first isolated from Zea maize or corn kernels. that is why it is called as zeatin. It is called as zeatin. Okay, got it fixed into your brains? Yes. Let's move on to the second very most important point. Cytokinins are the derivatives of adenine and purines or purine. Adenine and purines. The cytokinins are the derivatives of purines and adenines and purines. Remember this, very important. Auxins are the derivatives of indole acetic acid. Gibbsellins are the derivatives of acids. Remember that, gibberellic acid. These cytokinins are the derivatives of adenine and purine. Okay. Now, these cytokinins are naturally synthesized in the regions of rapid cell division since they have specific effects on cytokinesis. It's very important and it's very easy to remember. So those are the important points on cytokinins. Now we will be moving on to the physiological effects. We have only some 2 physiological effects. You have to remember and you will be remembering it very simply. Let's move on to the physiological effects. The first physiological effect is apical dominance. You have already said about this apical dominance in auxin, and the same thing is done by cytokinins. No, they overcome apical dominance. They help in overcoming apical dominance. If you spray cytokinins on any plant, the apical dominance can be overcome. Now there are 2 methods for overcoming the apical dominance. One, spraying cytokinins. 2, cutting the apical bud. If there is a shoot, just remember this is the apical bud. If you remove this apical bud, apical dominance can be overcome. Then there is a chance of lateral buds coming out. Apical dominance can be overcome in 2 methods. What are they? Spraying cytokinins or using cytokinins, or by removing the apical bud. Okay, like this apical dominance can be overcome. And the last and the second physiological function for this video and also cytokinins is they promote nutrient metabolism. They promote nutrient metabolism and help in delaying, I have already said, delaying leaf senescence, leaf folding. They delay leaf folding. See, nutrient metabolism, we have already seen in the biomolecules, nutrients are mobilized to the younger parts from older leaves or senescent leaves to younger parts, so that these younger parts grow, and they promote this nutrient metabolism to occur rapidly, and therefore they can delay leaf senescence. They can delay leaf senescence, younger leaf senescence. So that's it. From senescent parts, already older parts, they promote nutrient metabolism. They take all the nutrients to the younger leaves, so that nutrients are available. Then how the leaf can get old? Right? So that is how they help in senescence, leaf senescence, delaying leaf senescence. So next video will be on ethylene, gaseous PGR, and that's it for this video. If you like the video, please hit that like button very hard and also subscribe to our channel BioLab. This is VAMSI signing off from BioLab, and please do not forget to attend the test and also purchase the notes. Thank you guys. Have a great day further.